Welcome to Genomics Now, a podcast series where you can learn how genomics is developing in England's NHS. This podcast series is recorded in 2024 and is part of the North Thames Genomic Medicine Services Educational Toolkit. In each bite-sized episode, we explore what is changing in genomics. We answer the questions on genomics we've been asked by the different healthcare professionals. We talk about the ways genomics may impact different healthcare roles and how genomics can be used to successfully improve patient care. Hello, I am Dr. Shazia Mahamdallie, and I'm part of the North Thames Genomic Medicine Services Workforce Education and Training team. Today I'm back with Dharmisha Chauhan, the Lead Pharmacist at the North Thames Genomic Medicine Service Alliance. In our conversation today, we explore how research has facilitated the introduction of pharmacogenetic testing into clinical practice. So Dharmisha, hello again, and it's great to have you back.
Hi, hello.
So Dharmisha, I'm a research scientist by background, so a champion of research studies and clinical trials. They play a crucial role in advancing clinical medicine by providing the evidence needed to evaluate safety and effectiveness of new treatments, therapies, and diagnostic tools. So can you kick off this episode by explaining to us what types of trials and studies are most often used for evaluating the efficacy of new clinical interventions in your field of pharmacogenetics and medicines optimization?
Yeah, so to implement anything into routine clinical care, we need evidence. And within healthcare, for any given intervention, especially a new interventional treatment, we consider randomised control trials as a gold standard approach. And it's considered a gold standard because the randomisation is balancing out the participants and it gives us the ability to minimise biases. So within randomised control studies, an intervention will generally be compared to a standard of care approach, which in some cases could be no intervention at all, such as a placebo. With respects to pharmacogenetics, we tend to see patients being enrolled in two different arms. So one would be the genotype guided arm versus the non-genotype guided arm. And this can be in the form of testing the role of a panel test or to obtain more in-depth data of a particular gene pair for a specific population. So, for example, the role of CYP2C19 testing for children being treated with voriconazole, which is an antifungal treatment. So in contrast, a cohort study is really a type of observational study, and this can be retrospective or prospective in design and allows a group of people who are interested in where you can follow them over a period of time. And it can include one or more groups where one group is exposed or had a particular intervention, or you can also compare a low risk group to a higher risk group. It's not the gold standard approach, but it can provide really useful data and signals on where you can expand a research question further.
Thank you. What's interesting about the conversations that we've had is that some believe pharmacogenetics shouldn't require the same rigorous standards as other targets in their treatments. What are the arguments for and against this view?
Yeah, so there are differing views, and some feel that pharmacogenetic testing, where we are using the result to adjust dosage or identify for an alternative treatment should be prescribed, shouldn't really be subjected to these higher standards because we should really consider it as a safety test, which would be no different to how we would check a patient's full blood count or biochemistry before we prescribe a drug. So this thought is, especially with drugs where there is a really strong pharmacokinetic or pharmacodynamic data evidence base, that a certain metabolizer status, for example, would lead to greater side effects. So, for example, if a drug is given to a poor metabolizer and it would lead to side effects, which in really severe cases would lead to hospitalization. But others feel that we should have robust data before implementation, especially when we don't have the available pharmacokinetic or pharmacodynamic data, which is in fact becoming more frequent for drug studies because they don't really focus on this after the early phase studies. And so when it reaches phase 3 level, where you'd probably see it done in a randomized control setting, they really stop this kind of data collection.
Debate that will continue on, no doubt. So throughout this series, we frequently use clopidogrel as an example to illustrate pharmacogenetics. Dharmisha, Could you share how research on clopidogrel has generated valuable data that's led to the introduction of pharmacogenetic testing and influenced its prescribing practices, say, for example, in cardiovascular care?
Yep. So as we discussed in episode 2, clopidogrel is a prodrug prescribed within stroke and other cardiovascular conditions and needs to be metabolized by enzymes into its active form. And CYP2C19 is the main enzyme involved. And those that cannot metabolize a drug, e.g. poor metabolizers, may be at risk of reduced clinical efficacy. So let's focus on the data available for the role of clopidogrel in stroke as an example. So one of the earliest studies was conducted by a research group in China, and they published their results in 2013. And they recruited 259 patients who were diagnosed with ischemic stroke. And patients had pharmacogenetic testing done for CYP2C19 as well as a range of other genes. And this wasn't a randomized control trial, but it was a prospective cohort study where patients were followed up at 7 days, 3 months, and 6 months. And the primary outcome was a recurrence of stroke. The results indicated that CYP2C19 patients who were poor metabolizers did have reduced antiplatelet response, and that was compared to normal or ultra-rapid metabolizers. However, there was no control arm, and the primary outcome wasn't really well defined when you go through the study, and the follow-up of 6 months was quite short. So since this paper, there are several other studies that have been published, and predominantly they are from China. There are a few from the United States, and the majority of these are based as cohort studies.
Yes, and what you just described there was from 2013 from China. So more up-to-date UK data, for example.
Yeah, so in 2021, there was a UK paper published by Dr. Luke Pylin and his team, and they assessed 7,477 UK biobank participants. And of these patients, they found that 110 of them had CYP2C19 testing done, and they were also treated with clopidogrel. And these patients aged between around about 36 to 79 years old. And when they had their first clopidogrel prescription, and they were on treatment for around about 2.5 years, or 2.6 years to be precise. And their primary outcome measure was inpatient diagnosed ischemic stroke, myocardial infarction, or angina. And from this data that they found, the team identified that 28.7% of the participants were poor metabolizers and subsequently had a higher rate of ischemic stroke. And so they use this method called life table, which is new to me, but it's a statistical tool to obtain survival patterns over a population over a period of time. And using this method, they found that CYP2C19 poor metabolizers had a 22.5% risk of developing an ischemic stroke compared to 15.4% who were not poor metabolizers, thus indicating a 7.1% excess of risk of stroke. And then from a systematic literature review, it was conducted by Dr. John McDermott and his co-authors in 2022. They, in fact, identified 28 studies overall conducted within stroke. And what they found out that most of the studies are retrospective or prospective cohort studies in design. But overall, the analysis showed that there was a value in CYP2C19 testing, but specifically more in the Han Chinese populations. So what this literature review and with the data that we have from UK populations, it shows that we do need more data to be generated, which is representative of the UK populations in a prospective manner, ideally, particularly as our population is very diverse.
Yeah, that biobank data is really, really helpful, but it does need to broaden its diversity. But that's a real jump from a couple of hundred people in the China study back in 2013 to over 7,000 now. So in the example that you just described there, stroke patients undergo genetic testing for CYP2C19 gene, which helps determine the amount of the appropriate prescription, I should say, of clopidogrel. So this illustrates a study focused on a specific gene within a specific patient group. So can you share an example of a study that has generated data through gene panels? So that means analysing multiple genes simultaneously.
Yep. So there was a study that I would consider as a landmark study. It was published in 2023, and it's called the Preemptive Pharmacogenomic Testing for Preventing Adverse Drug Reactions, or we can call it PREPARE for short, which is how I'll be using the terminology. So PREPARE was a very large and hugely ambitious study. It's a multi-center European randomized control clinical trial. And they recruited 6,944 patients. And they were enrolled where 48% were in the genotype-guided arm. And this multi-center study included 7 European countries, of where Liverpool was one site. And the aim of this study was to investigate the clinical utility of a preemptive genotyping strategy approach using a pharmacogenetic panel, as this really hasn't been rigorously assessed before. And the participants or patients, they were genotyped for 50 germline variants across 12 genes, and they all had an actionable variant in terms of dosing guidelines that are available. And they tested around it represented 43 drugs. So I'm not going to go through all of the 12 genes and 43 drugs, but as a snapshot, they tested for CYP2D6, CYP2C19, CYP2C9, and CYP2B6. And those genes help guide prescribing for antidepressants, antipsychotics, anticoagulants, and pain medications as an example. And so the patients were randomized into 2 arms. A genotyping arm was the intervention group, and it was compared to a standard of care arm. So that was treatment as usual. So it should be noted that the treatment as usual group, or the standard of care arm, the participants were still tested, but the results were not used to guide treatment. So as a headline kind of results, it demonstrated that pharmacogenetic testing to guide prescribing reduced the incidence of new adverse drug reactions by 30%, which means that there were fewer adverse drug reactions in the genotype-guided arm. So really this indicates the benefits of preemptive testing. But it should be noted that the 30% reduction in adverse drug reactions in the study, it primarily relates to a reduction in the incidence of new adverse drug reactions within the timeframe of the study. But overall, in terms of the limitations, we do need to remember that both groups did have pharmacogenetic testing done. But overall, the study shows how a large pharmacogenetic panel can be adopted into prescribing practices. Despite some limitations, it is an impressive study conducted across different healthcare models and sectors across Europe. With respect to UK practice, this paper really makes me reflect on questions such as how large should our UK panel be if we were decided upon a multi-gene panel approach, and which drugs, should be considered using this approach.
There's a lot of thought that needs to go into that step change from one drug, one gene, to multiple drugs, multiple genes. But the potential is there, as you've just described. So, as you know, well, we like to end our episode on what's next. So what do you see is next for pharmacogenetic research studies? What are the gaps that you're seeing?
Yeah, I think there are quite a few research gaps within pharmacogenetic research in general. So I think that we need to look at the implementation of point-of-care tests across different healthcare settings, because there's a new technology coming through, and it'll be good to get data across different health settings and how it affects pathways. I also think that ancestry is a really big research gap as well, thinking about what variants are found in people of different ancestries and how that compares to the database that we have, which is predominantly that of European white Caucasian ancestry. It'll also be really interesting to look upon, and I mentioned this in the prepare element, the role of adherence and of medications if pharmacogenetic testing is done, and how patients report side effects. And, for example, if they had pharmacogenetic testing done, does this mean that a patient will follow their treatment plan that is being prescribed more closely compared to someone that doesn't? And how does that relate to how they report back side effects to their clinician? Also, a lot of economic or pharmacoeconomic data is missing, especially within a UK or NHS system model approach. So, for example, if you are implementing pharmacogenetics, then what economic impact does that have, especially on elements such as the unintended consequences? And also, if you were picking a different drug, would that alternative drug have higher risk associated with it? And, what does that all mean? So essentially, it means that we really need to get more people involved in pharmacogenetic research, especially pharmacy professionals, because it's all related to medicines. So yeah, that's what I think.
I'm a researcher at heart, so I agree with you there. So thanks, Dharmisha, for another great episode.
Yeah, so thank you. And hopefully this gives, our listeners a snapshot of how pharmacogenetic testing can be studied. And we'll also provide links to the papers that I referred to as well.
Oh, that'd be really helpful. Thank you so much. Thank you for listening to this episode of Genomics Now, a series of conversations hosted by healthcare professionals. The North Thames Genomic Medicine Service aims to complement other genomic-focused educational tools with these bite-sized podcasts. So do visit our website for more resources and updates on genomic-based clinical developments and research. If you've enjoyed this episode, don't forget to share your thoughts and spread the word by tagging the North Thames Genomic Medicine Service on social media.